Spermidine, a naturally occurring polyamine found in certain plant and fermented foods, has become one of the most intriguing compounds in the study of cellular rejuvenation and longevity. Unlike many nutrients that serve primarily as building blocks or cofactors, spermidine acts as a signaling molecule, directly influencing genetic and enzymatic pathways that regulate cell maintenance and repair. One of its most remarkable effects is its ability to induce autophagy, a process crucial for cellular renewal, by inhibiting EP300, a key enzyme that ordinarily suppresses autophagic activity. When EP300 is inhibited, the molecular machinery that initiates autophagy is unleashed, enabling cells to recycle damaged organelles, especially worn-out mitochondria, through a specialized form of autophagy known as mitophagy. This not only preserves energy efficiency and metabolic balance, but also helps delay age-related cellular decline. The unique appeal of spermidine lies in its ability to mimic some of the effects of caloric restriction, one of the most validated interventions for extending lifespan in model organisms. Like fasting or reduced caloric intake, spermidine influences longevity pathways involving SIRT1, AMPK, and mTOR, yet it does so without requiring dietary restriction. This positions spermidine as a compelling candidate for nutritional strategies aimed at health span extension, offering the benefits of cellular cleanup and metabolic recalibration in a way that's sustainable and accessible. In both animal and human studies, spermidine has been associated with improvements in cardiovascular health, enhanced immune function, and even increased lifespan, potentially through its broad activation of autophagic and mitophagic responses across various tissues. One of spermidine's most critical contributions is its role in mitochondrial maintenance. As we age, mitochondria, the energy generators of the cell, tend to accumulate mutations and functional defects. By stimulating mitophagy, spermidine encourages the removal of these underperforming organelles, paving the way for the generation of new, more efficient mitochondria. This process not only preserves metabolic health, but also reduces oxidative stress and inflammation, two core drivers of degenerative diseases. Moreover, emerging research suggests that spermidine may support genomic stability, reduce DNA damage, and improve protein homeostasis, factors that contribute directly to slowed cellular aging. From a nutritional standpoint, spermidine is found in a variety of whole foods, though concentrations can vary. Wheat germ is one of the richest natural sources, offering a potent dose along with complementary nutrients like vitamin E and B vitamins. Aged cheeses such as Parmesan also provide meaningful amounts of spermidine, thanks to the fermentation process that concentrates polyamines over time. Mushrooms, particularly shiitake and maitake, contain moderate levels and contribute additional antioxidant and immunomodulating compounds. Legumes, soy products, and certain fermented vegetables like natto may also supply smaller, cumulative doses. A dietary pattern rich in these whole foods, especially when consumed consistently, can gently elevate spermidine intake and support its cellular effects. In addition to food-based sources, spermidine is now available as a supplement, often derived from purified wheat germ extract. These supplements offer a more concentrated and standardized dose, which may be useful for individuals seeking targeted support for aging-related concerns, especially when dietary intake is inconsistent. Preliminary trials in humans suggest that spermidine supplementation is well tolerated and may improve markers of cardiovascular health, memory function, and immune surveillance, though long-term studies are still ongoing. Importantly, spermidine appears to work synergistically with other health-promoting behaviors, such as intermittent fasting, exercise, and nutrient-dense eating, by reinforcing autophagic signaling and enhancing the body's capacity for self-renewal. Perhaps most encouraging is the way spermidine integrates into the broader framework of longevity science. Rather than acting as a blunt pharmacological tool, it works subtly and systemically, supporting the body's own intelligent repair mechanisms. It doesn't override the cell's natural decisions about when to recycle and when to grow. It simply makes those decisions more efficient and timely. In this sense, spermidine represents a shift in how we think about aging interventions, from suppressing symptoms to reactivating the core processes that keep us resilient and biologically youthful. As our understanding of autophagy deepens, It becomes increasingly clear that sustaining this internal renewal process is not merely about fasting or reducing calories. It's about building an environment in which the body feels safe and supported enough to repair itself. Spermidine, whether through food or supplementation, is a powerful ally in this pursuit, gently guiding cells back into balance and setting the stage for healthier, longer living. Through small daily choices, adding wheat germ to yogurt, including fermented foods in meals, or simply supporting gut health to enhance polyamine production, we participate in a process that echoes across every level of biology: clean, restore, and thrive.